Hey everybody, welcome to part 3 of chapter 4. So we're going to talk about how to obtain a pure culture for growth in laboratory conditions. So a pure culture, definition-wise, is a population of cells that arose from one single cell. And this allows us to study that organism. In nature, organisms behave differently than they do in a laboratory, and very few organisms are actually cultured in laboratories. There are thousands of different bacterial species, but very few are able to be grown in a lab. And, well, I should say this first, but those are only the organisms we know. There are probably hundreds of thousands of other microbes that we don't even know yet. And so those organisms that we are able to grow, we want to study so that we can determine how they work, what nutrients they require. Do they cause disease? Can they be beneficial to biotechnology? And so we obtain pure cultures using aseptic technique. At this point in lab, you should have an understanding of aseptic technique. Aseptic technique is just the ability to transfer microbes or to work with microbes without contamination. So we minimize accidental introduction of other microbes to the original culture. And so we can grow microbes in nutrient broths, or on agar plates, or in gel. You have to use specific containers for growing microbes. You have to have certain types of media, and you have to have aseptic conditions. So if you have all of these, then you can take a single cell and grow it on a plate, an agar plate, and in approximately 24 to 48 hours, you will have a colony, and that colony is going to be at least 1 million cells. And then you can see that on the plate. So agar comes from algae, and it is used to solidify bacteria, or not solidify bacteria, solidify media. Most microbes can't degrade agar where they can degrade jello or gelatin. So agar is not destroyed by high temperatures, and because of that, it can be sterilized. So we sterilize the agar using an autoclave. It is liquid anywhere above 95 degrees Celsius, and it solidifies at around 40 degrees Celsius. So as it moves from that liquid to solid, we can actually pour the agar into Petri plates that are sterile. And this allows us to have solid media. The method of producing a pure culture from a mixed culture is known as the streak plate method, which is what we typically use in microbiology labs. So this reduces the number of cells through a process of streaking and sterilizing and streaking and sterilizing. It is a simple method, and you start on one portion of a Petri plate. You take a sterile inoculating loop and you dip it into your mixed culture of bacteria. You then streak 1/4 of your plate. From here, you sterilize the inoculating loop, kill any cells that are on it, and then you move from this area that has thick amount of bacteria into the second quarter of your plate. And you typically move 1 or 2 times into the already streaked area, and then you streak the second section. You re-sterilize your needle again, and you move from the second section to the third section 1 or 2 times, and then streak that area. And then you sterilize your loop for a last time, or not last time, but for a third time, and you're going to move the bacteria from the third section to the fourth section. And then streak this area, sterilize your loop, and cover your agar plate and incubate it inverted for 24 hours to 48 hours, and you will hopefully have these isolated colonies. So we typically use pure cultures. No, not typically. We always use pure cultures as in our stock cultures. Stock cultures are often found in agar slants, and they can be refrigerated. The cells can be frozen for long-term storage. Oftentimes, we even lyophilize the cells for long-term storage. And the lyophilization is freeze drying them. So we grow bacteria in our laboratories, and they show a characteristic growth curve where they have 4 basic phases: lag, log, stationary, and then death. And so we're going to go through these stages, and then we'll get into the next section of our lab or our lecture, okay? So in the lag phase, this is when you initially inoculate a new plate or broth. So the cells are being moved from one medium to another medium, and they start this lag phase. The number of cells does not increase. They start synthesizing enzymes required for growth. And depending on the conditions, if there's a lot of nutrients, there will be a short delay. If there are very few nutrients, then there's going to be a longer delay. Once the cell's prepared, it will go into the log phase or exponential phase. So exponential phase is where the cells actually divide. You have replication of the DNA, you have the cell wall formation, you have lots and lots of divisions occurring. And so this is a very rapid phase where you are exponentially growing the bacteria. So you start with 1 cell to 2 to 4 to 8 to 16, 32, 64, 128, and it just keeps going. This is also the phase where cells are most sensitive to antibiotics, because if the cells are producing lots and lots of proteins for division, if the cells are replicating, you are utilizing the ribosomes. So ribosomal antibiotics that target ribosomes are going to be very effective here. If you're growing new cell walls, then anything that targets cell wall synthesis is going to be effective here. Peptidoglycan synthesis, that's part of the cell wall of gram-positives, of all, or almost all, but mainly gram-positives. Then we move into the stationary phase. Stationary phase is a phase where there are few nutrients left. So a lot of growth has occurred. So the number of cells that are replicating, so you're still replicating some cells, but the number of cells that replicate are going to be equal to the number of cells that are dying. The cells that are dying are releasing nutrients so that other cells can grow. This leads us to the death phase. The death phase is another exponential phase, but it's a much slower exponential phase. So cells die at a constant rate, just like they grow at a constant rate. and the number of living cells actually decreases. So there is a phase of prolonged decline where a small portion of a population may survive. Typically, these are going to be organisms that are adapted to detrimental conditions, so they can survive in those poor nutrient conditions. So where the bacteria are going to be located is going to determine how well they will survive. So think about bacteria on a medium. The bacteria on the edge of the medium are going to be able to gain more nutrients easier than the bacteria in the center. So think about if you are an aerobic species, you're in the center, the nutrients and the oxygen are going to be depleted much faster because you have cells all around you. Whereas if you're on the edge, you have less competition. This allows cells in the middle or cells on the edge to survive and replicate, while cells in the middle end up dying and giving nutrients to neighboring cells. I'm going to stop here on this lecture, and then we'll get into the different environmental conditions that microbes need.